And I will pass to the next speaker, Gaia Camisasca. Hi, Gaia.
Can you hear me?
Yes.
Okay. Thank you. Thank you, Simone.
For you sharing your presentation. Sorry for the delay. I'm responsible for that. So Gaia will tell us about gas-induced drying of nanopores. Please, Gaia.
Thank you, Simone, and good afternoon to everyone who is connected. Today, in this talk, I will show you some results that we obtained with Antonio Tinti and Alberto Giacomello over the last year, where we study a drying mechanism of nanopores occurring in presence of a single atom of argon. More in general, the study of wetting and drying properties of nanopores in presence of diluted hydrophobic gases is much interesting in many phenomena which takes place in biology, but also in technology. So I will start this talk by giving you a few examples where our study can be relevant. So I will start from biology. So this morning we have seen during the first session of this Congress that the functioning of a class of ion channel relies on the so-called hydrophobic gating, which is the mechanism by which ionic currents are blocked through the membrane by the formation of a bubble in the, let's say, in the portion, the hydrophobic portion inside the cavity of the channel, the biological channel. In this context, we can think about the general anesthesia, and in particular to noble gases, which do show an anesthetic effect. Ion channels, besides, are most likely targets for volatile anesthetics, and in fact, in the literature have been posed hypotheses which relates the potency of anesthetics with the blocking of ionic currents through the membrane, as well as hypotheses regarding the involvement of a bubble formation inside a channel as one among the many possible mechanisms of anesthetic action. Therefore, the study, let's say, of the effect of hydrophobic gases to the phase behavior of water confined in this nanoscale appears to be very important in biology, but so it is also in the context of more technological fields. So yesterday, in the introduction, in the lecture given by Professor Casciola, he mentioned several type of devices which are based on nanoporous material. So, for example, he mentioned molecular spring, dampers, many others. But today I wanted to show you one more application, which we were discussing recently with Alberto and Antonio, which is the case of the reverse phase liquid chromatography. The reverse phase liquid chromatography is an experimental technique that allow you to separate analytes. So in this, typically you have a mixture of analytes in water, and you make them flow through a column, chromatographic column, which contains a stationary phase, which is an hydrophobic nanoporous material. Then, depending on the specific physical interaction between your analytes and the stationary phase, the different analytes will flow through the current at different velocity, and therefore we will be able to separate them at the end point of the column. So here, in this context, the drying of the nanoporous material contained in the column is a big problem, because it decreases the surface area exposed to the analytes. So basically drying deteriorates the performance of your apparatus. On the other hand, the experimentalist which uses this technique, they know that they can do something to slow down the drying of their nanoporous material, which is the fact that before starting the experiment, they gas water. So, sorry. Okay. So in this context, also the effect of hydrophobic gas dissolved in the water seems crucial. So I will go on and show you our system. So this is in this picture. This is that we simulated. We have a cylindrical nanopore with a radius of 7 angstrom, which we carved from crystalline material, which is shown here in gray. Then the nanopore is placed in contact with liquid water, where we left two free interface, liquid vapor interface, which guarantees isobaric condition during the simulation. And in one of the two bulk, we dissolved only one argon atom, which is the red guy here. This particular pore that I'm showing you is hydrophobic, as we characterize it with a contact angle. So we started by running a bunch of free molecular dynamics simulation with a starting configuration which is shown here. So we have a pore which is filled with water, and the argon atom which is far in the bulk. And in view of the simulation, we could observe what is here called a successful event. So we could observe the atom being attracted toward the nanopore, and here you see the entrance of the argon atom inside the nanopore. You see the fluctuation of water inside the nanopore increases, and eventually it leads to the wetting of our nanopores. Just to give you a time scale, we observe that from the entrance of the argon inside the nanopore, the drying occurring around 2 nanoseconds. We also characterize a bit the situation while the pore is filled with both water and the argon atom. So here, for example, we are plotting with the blue curve the probability distribution function of the argon as a function of the radius of the nanopore. I remind you that the wall is located at 7 angstrom, so we could see that the argon atom, preferentially resides at the wall. On the other hand, we could also quantify the fluctuation of the number of water molecules which are contained in the pore. So while the pore is filled only with water, we obtain the red distribution, which is pretty sharp and peaked at around 72 water molecules. But when the argon is inside, as you can see, the distribution broadens, and also this, let's say, rises a component at low filling value, which are the fluctuation that eventually lead to the wetting. We also wanted to characterize the kinetic of the process, and so we performed some free energy calculation. We applied restrained molecular dynamics with 2 variables. The first variable is the number of water molecules contained in a control region, which is this rectangle, blue rectangle here, which is a region enclosing the pore. And while the second variable is the position of the argon atom along the pore axis, which is this data variable. We obtain, so this in the center, you can see the free energy map that, the free energy surface that we obtain. On the x-axis, we have the axial position of the argon. The white vertical line marks the entrance of the pore, so everything which is right to the white line corresponds to position of argon inside the nanopore. While on the y-axis here, there is the number of water molecules inside the control region. As you can see, this free energy surface is mainly characterized by two horizontal valleys. One, the upper valley corresponds to state of, the wet state of the pore, and here we observe two main states, the state A and C, which correspond to the wet pore with the argon far in the bulk, and the state C to the wet pore and the argon atom inside. Correspondingly, for the bottom valley, which instead correspond to empty state, so dry state for the nanopore, we have the B state and the D state, where the argon is in the bulk or inside the nanopore. I also remind you that when I show you before the video of the free molecular dynamic simulation, we started a configuration which is of the type of similar to the A state, because we had the bulk, the argon in the bulk and the filled nanopore, and we end up in the state D, where there is the bubble and the argon atom inside. The D state is also the stable minimum in this free energy landscape. We then focused on two main paths on this surface, which are shown here in red and black. The red path corresponds to a path where we first observe the drying of the nanopore, and then the infiltration of the argon, moving from D to D to D, correspond to the infiltration of the argon inside an empty nanopore. The black path instead is kind of the opposite path, because we first observe the infiltration of the argon particle inside a wet nanopore, and then in presence of the argon, we observe the drying by moving from C to D. Now I want to show you in this other plot how the free energy changes along these two different paths. So the color code is the same. It is the same in the left and right figure. So we let's first focus on the red path. Here, as you can see, from moving from A to B, we first see the drying of the nanopore, which is a process which is characterized by a barrier of 2.5 kVt. Once we overcome the barrier and we arrive to the state B, then the argon atom, can enter the nanopore with a process which is barrierless. So without a barrier, we can move from B to D. Now let's see the black curve, where we first note that also in this case, so we can see a barrierless event. So the argon can enter a wet pore again, still without any barrier. But now this is the nice result that we obtain. Once in the state C, we see that now the drying of the nanopore happens with a vanishingly small drying barrier. So basically, therefore, with only one argon atom, we could abate the drying free energy barrier of our nanopore. So these results we tried to study this mechanism, so that we found also for other values of hydrophobicity. So we did some more simulation. In particular, we also studied two more nanopores. One which is characterized by a contact angle of 93 degrees, so it is a pore which is almost neutral to wetting, and then a third more hydrophilic pore with a contact angle of 85 degrees. As you can see here, we basically obtain the same phenomenology. In particular, we in both the case of the neutral and the hydrophilic pore, we can observe that the drying in presence of the gas occurs with a barrier which is smaller than the drying barrier when there is not the gas inside the nanopore. So I finished to present some results, and now I go to the conclusion of our study. So to summarize, first, we observe a mechanism by which a gas particle is hydrophobically attracted inside a nanopore. And once it's inside, the gas particle catalyzes the drying. In which way? By decreasing the free energy barrier. In the case of our nanopore, our hydrophobic nanopore, we actually saw that one single atom is enough to completely abate the drying free energy barrier, and this will also be true for pore which are of size comparable to the pore that we investigated. So basically for sizes which are also comparable to the typical intersection of biological channel. These results, besides, also support a possible mechanism for the action of volatile anesthetics, which involves hydrophobic gating, with the anesthetic molecule or atom that once is inside the pore domain would favor the formation of a bubble. Besides, I also want to remind you that our nanopore is a model nanopore, so the mechanism that we observe is quite general and specific, which would indeed also explain the variety of hydrophobic gases, which serve as general anesthetic. And finally, I would like also to link back to the technological application. Our study showed basically the mechanism behind something that was already known from the experimentalist, which is the importance of controlling the amount of dissolved gas in the liquid water. The liquid water may, of course, also suggest the usage of the gassing protocols in other fields where they are up to date not used still, and of course, it can also inspire new technology. For example, we can think about other fields where the drying can be functional to the technological application, such as, for example, a fast recovery of dry states of your nanoporous material. So I would like to thank you all for listening. Of course, I want to thank Antonio and Alberto, which are co-authors of this study, but of course I want to thank them together with Carlo for organizing this night's conference, and also Silvia and Flavio, which I know they worked a lot to make it possible. Thank you.
Thank you to you, Gaia, for your nice talk.